Okay, yeah. So it's recording right now. Um, hmm,
Yeah. Oh, do you have the slides? Oh, I think I sent them to Noli and not to you.
yeah, they might be in
Yeah, sorry.
Dropbox. Yeah. Mm.
Part of the project still, I'm guessing. Yeah.
Hmm.
What's that? Never too small. Yeah. Well, thanks for showing up, even though it won't be a quiz or anything. I hope it'll be interesting to you guys.
Hmm.
Okay, I'm putting it in the group chat.
Mm-hmm. Okay.
Got it.
Yeah, it's here. You can download it or present however you want.
Oh, is it downloaded?
Oh, okay. Yeah, I can download
Well,
it.
we don't have to,
Uh,
as long as like your internet is like good or whatever. Oh no, don't download it because it's Google Slides, so
yeah, yeah, okay.
it'll,
Let me
if you
make
put it in PowerPoint, it'll probably feel like a little weird. So oh, now you're on Millie's line.
I am not sure what happened. No, I think this is my line.
Hmm.
Hmm.
But it had Millie's space there.
Oh, wait, no, I'm on Manoli's account.
And it's got his background. Yeah.
Yeah, yeah, I can check.
Oh, you're coming back at 2:30? Hey. Okay, I'm screen sharing now, right? Do we need the like speaker on in this room? It's kind of weird. It's like echoey. I mean, there's just us. Oh, it's just default. It's fine. All right, thanks for coming. Today I'm going to talk about metabolic modeling and modeling uh, networks in metabolism. So I'm going to start out and give you guys kind of an overview of some like really basic metabolism and biochemistry, because then we'll it's like in no in no way comprehensive, but we'll draw from that for some examples later on when I describe how how we're modeling these things. It'll help give some specific examples. Okay, so metabolism is really all of the chemical reactions that happen in a cell. So previously we talked about small molecules. Now we're talking about the systems of enzymes and intermediates that lead to the generation of those molecules, and primarily in living systems. And there's two main components in a cell. One is catabolism, that's breaking down molecules that come in for either reducing power or energy, or really just to convert them into other molecules that you can use to increase biomass and make more cells. And so that's the latter part, which is anabolism. That's building up biomass and producing new copies of yourself. So why would we model metabolism? Well, it's really the one of the core functions of any cell. So far, we've been focusing mostly on proteins, but the function of a lot of these proteins is really just to process these metabolites. So this is kind of like the raw material going in and out, and we can think of those proteins as being little machines that operate on those raw materials. Metabolism is the basis of a number of different diseases, and it's really a focus when we're thinking about synthetic biology and organism designing new organisms to carry out different functions. We have to be really cognizant of metabolism and how those new organisms are going to be processing the chemicals they see. And sometimes the biotechnology that we want to engineer is the production of different molecules. So many molecules are very difficult to make synthetically, and we rely on organisms to produce them. So then we want to try to guide the metabolism to produce more of the products we want and less of the byproducts we don't want. It's also helpful to, I should say, to model these things, because there's a lot of different control points that determine how much of each different metabolic pathway is produced, the products of each metabolic pathway. So when we model the entire metabolic network, it gives us a good idea of where those control points are, so that we know if we want to engineer like more flux through this pathway and less flux through that pathway, we can predict where which enzymes we need to intervene at to affect that change. All right, so some terminology: metabolites. These are the small molecules that we spent a couple of class periods on previously. Enzymes are the primarily protein-based components that serve as catalysts for metabolic reactions. Talk about pathways as a set of enzymatic reactions, one after another, although they can also branch, that produce some end product from some input set of substrates. And we'll talk about the network as the entire set of pathways in an organism, or maybe some subset of an organism, like the catabolic network or the anabolic network or the lipid biosynthesis network. Here's a view that I just took from Wikipedia that is really just a subset of all the metabolites, and showing the connections and how they're interconnected in these pathways. On the Wikipedia metabolism page, you can also find somebody made this into like a metro map, which is kind of cool. And you can like hover over that and kind of explore all the different pathways. But what you see is how they're all these different pathways are really connected together, but they're also very modular. I want to spend a few minutes going into central metabolism or central carbon metabolism. And so these are the pathways that process carbon. So most of the carbon coming into a cell, let's say it's coming from glucose or something like that, that glucose can be diverted into like a few different pathways. So we can use it for carbon to build up new biomass. We can break it down for energy, or we can say, hey, there's a lot of electrons here. We want to use those for reducing power, and that's also important for building biomass. So there's a lot of things we can do with that carbon that comes in. And central carbon metabolism kind of describes the key pathways that process like mostly 6 carbon sugars that come into the cell. Okay, so this is going to include glycolysis, pentose phosphate pathway, and the carboxylic acid cycle, the TCA cycle. So glycolysis. So if I can move this. Glycolysis is the pathway shown here at the right. And so what we see is it's a mostly linear pathway that goes from glucose, which is 6 carbons, to pyruvate, which is 3 carbons. So halfway through this pathway, that 6 carbon sugar gets chopped in half. Okay. And so there's one molecule going through the first 4 steps here, and then the later 6 steps, there's 2 molecules that are going through it, and we can see that it takes in 2 ATP in the beginning, and it produces 2 ATP in the after the molecule is cleaved. So because each of those 3 carbon units makes 2 ATP, there's a net gain of 2 ATP, right? Because we use 2 ATP at the beginning, but then we produce 4. So there's a net gain of 2 ATP, and there's also a net gain of 2 NADH. And so if we're thinking about like what are the things that we need to balance inside a cell, one is carbon, right? Like however many carbon atoms we take in, that's kind of what we can use, right? And we can expel some of that carbon as waste or as like CO2, or we can turn it into the building blocks of new cells. And then biological material tends to be fairly reduced. So it tends to be fairly electron rich. So if we want to make new cells, then we also need to take in a lot of electrons. And so those, every NAD is going to carry 2 electrons. And there's another molecule, NADP, which is very similar, takes in 2 electrons as well, and has about the same reducing potential as NAD. But this is one of the ways that the cell discriminates catabolism, which is breaking things down for electrons or for energy, and anabolism, which is building up, doing biosynthesis of new material. NADH primarily used for generating ATP and making energy for the cell. NADPH primarily used for anabolic metabolism or building up new biomass. So in glycolysis, we're producing NADH. So it's mostly about catabolism and energy production. Okay, and we're also producing pyruvate. Okay, pyruvate 3 carbons, and is the that's going to go into the TCA cycle, as we'll see later on, and that's the source of a lot of the different carbon compounds that end up making amino acids and other molecules in the cell. Okay, glycolysis is kind of the first step when we get a glucose molecule, and it's found in almost every organism. And from there, we can go on and do fermentation if we're under anaerobic conditions, or we can use the TCA cycle together with the electron transport chain to generate lots of ATP if there's oxygen around. Okay, so that's one pathway, right? That's going straight down glycolysis. That is mostly, let's say, catabolism, although it's producing some carbon intermediates as well. There's another path we can take, though, and that's a pentose phosphate pathway. So we can also put our glucose molecules into the pentose phosphate pathway. And the pentose phosphate pathway leads to NADPH for biosynthesis, and primarily building up new biomass, for example, fatty acids. And then as part of generating that second NADPH, it involves kicking off one of those carbons into CO2. And because we're releasing a CO2, that lowers the free energy of those products. So that drives the reaction forward, but it leaves us with a 5-carbon sugar. Fortunately, it leaves us with a 5-carbon sugar, which is the basis for our nucleotides, so our DNA and RNA. So pentose phosphate pathway is producing lots of DNA, RNA precursors, as well as NADPH for biosynthesis. And it kind of makes sense that those go together, right? If we're building new cells, we have to replicate the DNA. We also need that reducing power to just do biosynthesis, make new fatty acids, make everything like that. So they're sort of aligned, but we might need a lot more of the NADPH. We might need a lot more electrons reducing power than we need DNA. So then there's other branches of the pentose phosphate pathway that will take those 5-carbon sugars and put them back into 6-carbon sugars. Okay. So obviously you get less of them because you spit out some CO2, but you can run one branch of the pathway without the other, and that's really key. And we're going to come back to this when we talk about modeling different metabolic systems, because these are the decisions that the cell gets to make, right? We can conserve electrons, we can conserve mass, we can conserve all these things in multiple different ways. Okay, so if we take the pentose phosphate pathway and go all the way back to glucose, now we're generating NADPH, but we're not producing ribose. Okay, or we can run them together and use that ribose to make DNA or whatever we need to do. Yep. So this is an example of why biological pathways have multiple choices. And so that's a system of reactions that defines metabolism is underdetermined. So there's multiple ways to process that incoming material. And we're going to think about, well, what is the cell optimizing for, right? Maybe it's optimizing for an ADPH. Maybe it's optimizing for ATP, in which case we would just go straight through glycolysis, right? Okay, the last pathway in central metabolism that I'll talk about is the TCA cycle. So here we get pyruvate in, and we're going to generate a number of NADH, some FAD, and also an ATP. as well as 2 molecules of CO2. And then those 3 NADH, if oxygen is available, then we can use that to actually generate a ton of ATP. I think in total, including glycolysis, is something like 38 ATP can come from one molecule of glucose. So one function of TCA cycle is to generate that NADH, which is catabolic, right? And it's going to be used for ATP production. Another is just to generate all these intermediates, many of which are used in other biosynthetic pathways. A lot of these intermediates are the basis for different amino acids, as well as generating intermediates for fatty acid biosynthesis. Okay. So that's all I'm going to say about metabolism, because it gives us something to talk about later in terms of how these different things are regulated. So what are the types of metabolic models that are out there? Well, one that we'll spend most of our time on is stoichiometric models. And so these models are very simple. The only thing going into the model is how many is basically just the mass balance of all the different chemical reactions, right? So if I, take a glucose and a phosphate, and I put them together, and I get glucose 6-phosphate, that's a reaction, and it's predetermined. So all I'm putting in is like the mass balance there. Say if I have this this reaction and I list out all my metabolites, let's say one of them is glucose, one of them is phosphate, one of them is glucose 6-phosphate. I'm going to put like a +1 molecule of glucose 6-phosphate and a -1 molecule of glucose and a -1 molecule of phosphate. It's really that simple. So we can build like a giant matrix of all the different reactions that we have in our system and how they affect each of the different metabolites that we want to measure. And it turns out if we make some assumptions, we can actually learn a lot from those models. So they're very powerful. They're also very difficult to validate in a way. And I'll look at kind of what the state of the art looks like in terms of validating those models and things like that. On the other hand, we can do kinetic models, and this is usually something that is done when we have a pathway that has a relatively small number of different reactions. So here we can look at the individual reaction rates, and we can look at the enzyme kinetics. So if we know the concentration of metabolites in the beginning, and we know how the enzyme kinetics depend on those concentrations, then we can build detailed models over time. Of those kinetics and kind of look at the dynamics in the system. We can put thermodynamic constraints on our models. We can put these on any kind of model, but in particular for the stoichiometric models, if we're modeling a reaction that where the delta G is very low, then we're going to say that that reaction is essentially irreversible, and then we can say that the flux through that reaction has to be positive, right? Or we can put some bounds on what the flux through any particular reaction is based on the thermodynamic constraints. We can use ODEs to really get fine scale information over time for systems that are not at the steady state. Steady state is an assumption that we often make when we're using the stoichiometric models. And we can do genome scale models. So we can get an organism, usually like a microbe, sequence the whole genome, figure out what enzymes are there, and so what chemical reactions are theoretically possible, and then build a genome scale model for how metabolism works in that organism. It's pretty difficult to get measurements on the the amount of different enzymes that are there, and the amount of different intermediates that are there, or especially fluxes. So it's very hard to test those models, but we can use them to make predictions, and we can see how often those predictions are true. Hybrid models, like it sounds, can employ more than one. One useful one is we can use stoichiometric and kinetic models together, and people are starting to think about machine learning based models. Okay, so genome scale models are aimed at identifying dynamics in the entire metabolic network of an organism. So we can predict maybe not dynamics so much as fluxes. And based on those fluxes, we can put some constraints on the growth rate of an organism. And then we can perturb it. So for example, we can take a genome. We can say, like the genome for E. coli, we can grow it on a defined media where we know exactly, what sugars and amino acids and so forth are present in the growth media for these cells. And then we can look at the growth rate, and then we could say, okay, if we doubled the amount of glucose, how would that change the growth rate? Or we can say things like, what if we knocked out this particular gene? Would it still be able to grow? And if so, like how would that impact the growth rate? And that's one way to kind of test these genome scale models. And if they perform well, then we can start to use those genome scale models if we're trying to, let's say, engineer a new E. coli that is particularly good at producing some compound that that we want to extract out of a bioreactor. Okay, so how do we make a genome scale model? We need a genome, so we got to sequence the genome. We need to annotate the genome. Used to be done by hand. Now there's fairly easy to use pipelines that will go through, annotate, especially for microbial genomes, and do a really good job. So it'll identify all the different proteins. And then for the proteins that have some enzymatic function, it'll assign a number to those proteins, an enzyme commission number, an EC number, and that's a number in 4 parts of kind of increasing specificity, and it really defines exactly what that enzyme is doing, what its products and substrates are. Usually, even though the current methods for automatically annotating genomes are pretty good, especially if you're looking at an organism that's been annotated many times before, like E. coli, it still requires some refinement. So when I used to be doing a lot more of this, so annotating genomes largely by hand, in, let's say, the early 2000s, the complete pathway for biosynthesis of alanine in E. coli, it's like the sort of the most basic amino acid in the most well-studied organism, it was unknown. It's like we knew that E. coli could synthesize alanine, but we didn't know what all the genes were that were involved. Okay, those weren't discovered until I think the late 2000s, right? So and certainly there are enzymes even in E. coli that catalyze reactions, and we don't know what they are. Maybe not as basic as alanine biosynthesis, but certainly there are many, and there are many in non-model organisms. So we need to remove the false positives, and we kind of identify those as something that looks like it catalyzes a reaction, but none of the enzymes needed to make the products are there. let's say it's a long pathway with 10 steps, none of the, none of the steps to make the substrates are there, and then none of the enzymes that carry it to the, to the end of the pathway are there. So then we look at that and we say, well, this isn't very useful. This enzyme probably does something else. And then, in some cases, it's like, oh gosh, here's a long pathway, and it's a linear pathway. One thing goes in, one thing comes out, but there's a step missing in the middle. So we assume that, oh, that enzyme probably is there. We just don't know what it is. And so then we can add that. And that's all part of the refinement process when we're building a genome scale model. And that can help us, make hypotheses about what functionality the cell has and what set of enzymes it has, maybe that we haven't discovered. So there's a lot of tools that do this. Like I said, KEGG is really useful database of enzymatic reactions. And so we can blast against UniProt or KEGG or Metapsych, and those all have collections of proteins that are annotated with these EC numbers. And we can transfer those annotations onto the genome that we have. There's also some tools for taking a whole genome and doing this automatically. I'm a big fan of the seed tools, and so model seed is helpful in making automated annotations of enzyme function. Seed is a database that was it's been around for at least like 20 years, and it really started with getting everybody in the community who was doing sequence analysis. And those days it was, it was largely done by hand. So people who were experts in different enzymatic pathways and different enzymes to go in carefully and try to find different versions of those enzymes, across many organisms. So it's built on a database that had a lot of human knowledge in there, and they really helped to clean up some of the annotations. No, I think I kind of covered refinement. Okay, so once we have like a genome scale model, then before we use it to make predictions, we really want to validate it. And that's where things get like a little difficult. So we can compare our model predictions to growth phenotypes. So we can grow, let's say, our E. coli on different media, and maybe some media it doesn't grow. And ideally, if we had a good model of metabolism, it would tell us what media this particular strain of E. coli is going to grow on versus not. Or we can look at different gene knockouts, and we can predict which knockouts are actually going to inhibit growth versus which ones are going to allow it to grow freely. We can look at metabolic flux. So if we put in carbon compounds, let's say glucose, that's isotopically labeled, then we can go in with metabolomics, and we can look at how that isotopically labeled carbon makes its way into other pathways. Okay, and that tells us something about the flux. So if we assume the cell is at steady state, then the levels of each of those metabolites in the cell is going to stay the same. But if we're putting in a lot of isotopically labeled compounds, then the fraction of each of those metabolites that is isotopically labeled is going to start to increase. And we can see, like how it's increasing, and then identify like what's the flux through that pathway. And that's really what we're predicting when we make these genome scale models is the flux through the pathways, because we usually assume that the system is at steady state, and the actual levels of each metabolite at any given point in time are relatively constant. And we can, put some metrics on so we can make sure that the model is predicting sort of the growth rate of cells accurately, predicting the amount of energy that the cell has accurately. And then we can use those experiments also to constrain the models. So in theory, it works great. In practice, it works okay. But if we have a lot of missing genes, which happens in non-model organisms, that can be an issue. And of course, for large models, then it can take quite a bit of computation to solve these things. And generating high quality data to validate these models is difficult, just because there's so many different metabolites to look at. Okay, so I want to talk about how we model these reactions. So we'll take a very simple chemical reaction. So here we have A going to B at a certain rate. And the rate here we'll just call V1. Okay, so now we can express this reaction as like a very simple differential equation. So dA dt is going to be negative V1 because we're removing, some number of molecule A, and we're making exactly that number of molecule B, because we have 1A going to 1B. And so dB dt is positive V1. Okay. This might be our metabolic network inside a simple cell. But then to model it more completely, we need to think about, what's the flux going through the cell? Because I told you earlier, the levels of each metabolite, each intermediate in the cell, probably are fairly constant over time. So, but we know the cell is consuming glucose and it's consuming other nutrients. So we at least implicitly want to put those into the model. So we can think about a model that has a V0, and that rate might be controlled by how many transporters, let's say if we're bringing glucose in, how many transporters for glucose are on the surface, how efficient are they, what's their affinity for glucose, how quickly do they transport it across. All of those things will go into just one rate constant that we'll call V0. And we can also assume, let's say the volume of our cell is very small compared to the volume of the environment that it's in. And so when our cell takes up like a molecule of glucose, it doesn't really change the external concentration very much, right? So this is like an approximation. Obviously, over time, that's going to change. So if we looked at our cell over a very long period of time, V0 might not be constant, but so we can think about the time scale on which a simulation like this might make sense. And then, depending on what we do, we're probably going to have some waste products. So even if we utilize all of that glucose and turn it into ATP and release it all as CO2, like that, that's still a waste product, right? That CO2 is still leaving the cell, and so we need to model that as well. Okay. So we have a number of different frameworks for modeling these reactions. I showed you a simple one, an ODE framework, where we just look at all these rates and we build up our set of differential equations. We can model those rates very simply, or we can take extra things into account, and we can make those rates dependent on things like the specific concentration of metabolites and things like that. Stoichiometric modeling, that's kind of the easiest. Once we know the set of metabolites and the set of reactions, then we kind of have our stoichiometric model just by looking at the mass balance in those reactions. And then if we're talking about things that are happening, let's say inside a single cell, sometimes we have very small numbers of molecules. Okay, and this is something that somebody told me once, and they worked it out, and it was like approximately true. But it sort of blew my mind thinking about, like, what are the numbers of things that are like present in a cell. And so if we think of a cell at pH 7, right, that means it's got a bunch of water molecules, and it's got a few hydronium ions. It's got some, like, positive ions. It's got some hydroxide negative ions. And so it's got these free protons floating around the cell. And they're moving quite fast because they're in these networks of water. But at any given time in an E. coli, how many protons do you think are floating around when it's at pH 7? And we see, like, protons are being used in like many of these metabolic reactions. Jordan, how many protons in an E. coli at pH 7? 6. There's six in the cell at any given time. But they move really fast, and they can be used in these reactions. But the point is, like, we're talking about a single cell, talking about small numbers. And so things happen stochastically that where the behavior starts to deviate from what you would predict if you were modeling everything with differential equations, right? When we're modeling things with differential equations, we assume that there's so many molecules that it really follows these smooth curves. But when we're talking about things happening in one E. coli cell, especially if it's something like transcription or translation, we're talking about numbers of molecules that you can kind of count on on two hands. And so in order to get accurate dynamics in a system like that, we need stochastic models that can deal more fluently with those small numbers of molecules. So we'll talk about those. We'll dive into both the genome scale models. We'll talk about flux balance analysis, and then we'll go into the stochastic models. And there's many other types of metabolic modeling that you guys can look up if you're interested. Okay, I do want to say, before we get to the point where we're abstracting all of these reaction rates as like a single number, I want to kind of dive into what really goes into a reaction rate. Okay, so the general form of a reaction rate is that we have some constant, some reaction constant. Let's, for the moment, assume that that's K. It's just this rate constant. And if we have a reaction like the one at the bottom, or if I can move this, you can see. Yeah, I can. Okay. For the reaction A plus B goes to C, then we're going to have a reaction rate that depends on A to the first power times B to the first power. If it was A plus 2B goes to C, we'd have like our constant times A to the first power times B to the second power, right? So that's just our law of mass action. Is it's the product of the concentration of all the products determines the forward reaction rate. Okay. And of course, we may have a reverse reaction rate, right? Okay. But that rate is not necessarily constant. So let's say we have a reaction like A goes to B. We want to know that that reaction rate. Obviously, it depends on the concentration of our substrate. Here, I'm calling it S. But really, the more substrate that we put in, the faster the reaction is going to go. Why is that? Well, the substrate has to bind to the enzyme, and only when the substrate is bound to the enzyme can the enzyme catalyze that reaction whereby the substrate becomes the product, right? So if I have one enzyme in my cell and I have one substrate, that reaction is going to go very, very slowly because it's going to be a long time before that substrate finds its way to the enzyme, right? If I have a lot of substrate in the cell, so that in the local environment of the enzyme, everywhere the enzyme looks, it's surrounded by substrate, then one of those substrates is going to bump into the enzyme, it's going to bind in the active site, and it's going to convert. So in fact, these rate constants, when we look at them a little closer, they're not actually constant. You know, they really depend on the concentration of the substrate. And the rate of reaction not only depends on the concentration of a substrate, but I can make this same argument for every enzyme that's in my system. So at any particular concentration of substrate, if I have 10 enzymes, well, that reaction is going to go at 10 times the rate that it would if I only had one enzyme, right? So this is a really important point that sort of justifies the genome scale models that we're making, because by altering the gene expression, by a cell deciding, oh, which genes am I going to upregulate and which genes am I going to downregulate, it can upregulate the number of enzymes in any particular reaction to speed up that reaction, or it can downregulate it to slow it down. And it might do that via gene expression. It might do it by feedback loops where the product of the reaction binds to the enzyme and turns it off. There's a number of ways for the cell to do that. But one of the assumptions that we're going to make later on is like, oh, the cell is optimized to produce biomass, and it's going to optimize these reaction rates. And it's not immediately, at least to me, it was never intuitive. How is the cell going to change the reaction rates? Well, the reaction rates really depend on how much enzyme there is. They depend on how much substrate there is. Probably the substrate levels for most substrates are fairly constant, right? But they also depend on how much enzyme there is and how effective those enzymes are. And that is something that the cell can control. So if we assume things like, oh, in these conditions, the cell is optimized to produce ATP. In these conditions, the cell is optimized to produce new biomass. We can say, oh, what do we predict the fluxes through each of these metabolic pathways are in order to optimize biomass production, or in order to optimize ATP production? So that's going to be the basis of the genome scale modeling that we're talking about. ODE modeling, it's kind of like it sounds. We have, let's say, a simple reaction network here. Substrate 1 goes to substrate 2 to substrate 3. That should probably be product 3. And there's 2 reaction rates. And so we can build a set of a system of ordinary differential equations to model that. And the nice thing about that is we could start with just a bolus of S1, right? Maybe we're gonna like put the cell in, you know, from wherever it is into fresh media, and that media has like a bunch of S1, and it didn't have any S1 in the media where it was growing before. And so we know it's not at steady state. We could model how that S1 is getting converted to S2 and S3 over time if we kind of knew the rate constants, or especially if we dissected a particular pathway in vitro and we wanted to see, you know, how the substrate was converted to product over time. This would be a good way to measure that. Or it could be, maybe it's happening inside a cell, but we just induced the production of an enzyme, and previously the enzyme was turned off, we turned it on, and so, you know, the amount of different intermediates is going to change over time. Okay, so that's kind of one extreme is we're going to look at how the rates of different metabolites are changing over time. The other extreme is where we say, well, let's make this assumption that the rates of intermediates, the rates of metabolites, not the rates, but the amount of metabolites over time doesn't change. How much flux is going through each pathway? So these are stoichiometric models. It starts with a stoichiometric matrix. That matrix, every row of the matrix is going to be a different metabolite. Every column of the matrix is going to be a different reaction, and the elements are going to be the stoichiometric coefficients. Okay, and I'll show you an example of what I mean by that. And the mass balance equation is just the stoichiometric matrix times the vector of reaction rates that's essentially going to be the change in metabolite concentrations. So at steady state, we assume that the change in metabolite concentrations is 0, and so then, you know, we want to figure out what are the reaction rates where S times V equals 0. Okay, so if you remember from linear algebra, that's basically the null space of the S matrix, right? That's going to define what are the sets of reaction rates, like how much flux there is through each reaction in our network that is not going to change the concentration of any of the metabolites in the system. So now we've taken this whole big genome, and you know, we've done the mass balance, we filled in like all the equations, and now we can just solve this fairly straightforward linear algebra problem, and it's going to tell us, oh, what are the possible fluxes, you know, through this network. Okay, so we're going to look at a system of reactions, and here we're going to, again, we're assuming that the concentration of, you know, A, B, C, D, these are all constant. And so instead of having a complicated rate constant that depends on the concentration, things like that, if we assume it's constant, we can just take each rate constant to be a constant, like a number, right? And I should maybe refer to it as a flux rather than a rate constant, since that has a specific meaning. I'm just talking about how quickly each of these reactions is happening, which I've denoted like V0 through V5 here. And you can see that I have some arrows going out for things that are being excreted from the cell that don't connect to anything, and an arrow going in. Okay, these you know, we can put some limits on what they are if we want. but we can choose them to make sure the things in the middle satisfy the idea that none of the concentration of these things is changing. Okay. So our key equation here is our stoichiometric matrix times V, and V is a vector telling us how much of each, how much flux there is through each reaction. What is the rate of each of those reactions? S times V is 0. So no, the concentration of things are not changing. We're not looking outside the cell, so we don't care about those changes. And there's two scenarios, right? Well, there's actually three. We might have a determined system. So we might have as many metabolites as we have independent chemical reactions. And so there might be one answer, right, to how the system works. We could be overdetermined. We could have, lots of different reactions compared to the number of metabolites. And then we kind of have to like cross our fingers and hope that like there's no contradictory information there and that we still get one answer. But in reality, or at least practically in biological systems, these things are almost always underdetermined. Okay. So our system is underdetermined when the number of reactions exceeds the number of metabolites. And so our system has a number of different solutions that are all consistent with conserving the no change in the number of individual metabolites. So we can't uniquely solve for a set of reactions. We can't uniquely solve for that vector V, which tells us how much flux there is through each of those reactions. But we can define a space in which all of those V's are consistent with it, right? So then what do? Well, we have to choose what we think is the best point in that space, and then we're going to assume that maybe evolution has optimized the cell so that the cell has also solved that problem, and it also knows what is the best point in that space. And that's the basic idea of what we're going to do. So in E. coli, we can produce pyruvate through multiple pathways. And so our system is underdetermined. How do we get there, right? Could have gone straight through glycolysis. We could have taken half of our glucose and put it through pentose phosphate pathway, but pulled it back into glycolysis. We could have taken some of it through glycolysis, but then put the rest through pentose phosphate and used the carbon to make DNA. And so then we get less going through here. And so there's a number of different ways we can do it. Overdetermined systems in metabolic networks probably means you did something wrong with your modeling step. Honestly, it doesn't really happen in actual metabolic models, and unless they're that doesn't happen. Maybe if you make a bunch of measurements to constrain your model and your measurements have noise, and then you try to put those constraints on your model, you could end up with an overdetermined system. But if we're just talking about the stoichiometric matrix alone, it's not going to be overdetermined. Why are metabolic models underdetermined? Because we have a lot of redundancy in these pathways. So when we talked about pentose phosphate, even we said, look, if we want to make the same amount of NADPH as we're making ribose for nucleotides, then we only have to run half of the pentose phosphate pathway. But if we want the NADPH and we don't need the DNA, then we can go a different way, right? So there's multiple ways to get through this network, and that gives us a lot of redundancy. It gives us a lot of choices, a lot of places to control different aspects of the overall network. Okay, so how do we deal with it? Well, we're going to put constraints on, and then more importantly, well, maybe not more importantly, but importantly, we're going to define an objective function that we think is reasonable. And so two really straightforward ones that we've talked about. One is the cell is in growth mode and it just wants to make more biomass. The other is the cell is in a different mode and it wants to produce lots of ATP. Maybe it's undergoing a stress. Maybe it's fighting, doing chemical warfare with other cells in its environment, and it needs a lot of ATP to sense signals and respond and things like that. Whatever the objective is, we assume that the cell is probably fine-tuned to give a fairly optimal solution. So if we find the optimal solution, maybe it's close to what biology is doing. Okay, so we can look at central carbon metabolism. So glycolysis and pentose phosphate strongly overlap in their metabolite usage. So that gives us an underdetermined network, and we could come up with a solution either to maximize our ability to produce biomass, or let's say to make ATP, or our solution could be, hey, we want to generate twice as much NADPH that we're going to use to make new biomass as we generate NADH to produce energy, to produce ATP. So our constraint doesn't have to necessarily maximize one. It could cut a line between like 2/3 this way, 1/3 that way, and that relies on us to imagine what would the cell's best response in this environment be? What do we think it's been evolutionarily optimized for under these conditions? Okay, so flux balance analysis is basically kind of the mathematical approach that we take to account for this, and we can use this in to predict growth rates, how things grow on different conditions. We can use it to do metabolic engineering, figure out like, oh, which pathways do we need to interfere with to boost production of one metabolite or another, and that's been quite useful. We can also use it to study metabolic diseases. So, like, where would be the appropriate step to intervene at to eliminate some metabolic deficiency. So the workflow for FBA, again, it's a genome scale model. So we're going to do a genome annotation. Once we have that annotation, we get those EC numbers. Now we can look up those reactions in a database, and we can see what are the substrates and what are the products. And from that, we can build our stoichiometric matrix. We can set bounds for each reaction. So we can say, here's the minimum rate for this reaction, and here's the maximum rate. And we can use, kind of some plausible physical range for that. If the reaction is, if the delta G of the reaction is very negative, then we might say, oh, the V min is 0. Like the reaction can't go backwards, it's irreversible. So that would be an example of something. Or if it's, if we assume that outside the cell is very large and it has an infinite amount of glucose to provide the cell, and the cell wants to grow as quickly as possible, then clearly our network would just, suck in that glucose like infinitely fast, and our cell would grow very rapidly. But that's not realistic, right? There's some number of transporters on the surface of the cell, and there's some concentration of glucose outside. Those transporters can only operate this quickly, right? So then we'd put in a maximum bound on that particular reaction. So we can set up all of these bounds. We're going to include, explicitly interactions with the environment, although we won't model their concentration outside because we just assume they're not changing. So all we have to do is model the flux in and the flux out. And then we choose our biological objective, right? Once we have all of that, then we can just use techniques that optimize these linear problems. So it's basically a linear programming problem to find the optimal distribution of fluxes. And basically, the set of allowable fluxes is going to be a polytope in a high dimensional space. And one of the vertices of that structure is going to be, if it's a linear problem, one of the vertices of that structure is going to be have the optimal value, right? So we just have to find which one, and there are many tools out there to do this relatively efficiently. Okay. So our steady state assumption is S times V equals 0. Our linear programming problem is solve our system S times V equals 0, where our V's may be constrained. And then within those constraints, we want to maximize C times V. So C is going to be a vector of the same dimensionality as V, and it's going to tell us how to weight each of those reactions. So, for example, if I've got 100 reactions in the cell, and I want to maximize the production of NADPH, then my C vector is probably going to look something like it's going to have a +1 for every reaction that produces 1 NADPH, or a +2 if it produces 2 NADPH. And if it consumes an NADPH, it's going to have a -1, right? So I would have that vector. And now my C times V is, it's just going to take those reactions and weight them by, how much NADPH they make. So that would be like a really simple constraint. But if my constraint is making new biomass, well, then I want to produce a certain amount of NADPH, a certain amount of ATP, a certain amount of pyruvate, and I would have like all of these different metabolites that I want to make in a certain ratio, and then I would, that would go into my C vector, right? So this is how I would like put a constraint on it. Then once I have the constraint, then really I'm just calling like a linear algebra a library, and I define S, I define C, and I put in my constraints, and then I get a solution for what is the set of fluxes through these reactions that maximizes it. Okay, so here's an example for a simple system. A goes to B at a rate of V1, B goes to C at a rate of V2. So I can make my stoichiometric matrix. So my rows, again, are my metabolites, right? And then my columns are the different reactions. So in reaction number 1 for V1, I get -1 molecule of A and +1 molecule of B. and it doesn't do anything to C. But in the second reaction, I get -1 molecule of B, +1. Oh, sorry. There's a typo on the slide. That should be a +1 molecule of C, and that's going to define my my matrix, right? And then I'm looking for oh gosh. Oh, sorry, guys. I should have proofread these. My V vector should only have V1 and V2. There's no V3 in this in this simple network. Apologies. So that would be an example of my matrix. Let me just change. Can I even change this? Let me get out of this mode. All right, so let's change it here. Oh, I can't because I'm in Zoom or something. All right. All right. Well, this should be a +1, and there's no V3 here. Okay. So we have that's our V vector and our S matrix. We have our objective function. We're going to optimize our objective function. And so this, like we said, the set of feasible fluxes, so feasible V vectors, is just the null space of that stoichiometric matrix S, and it's constrained by whatever constraints we put on it. And linear programming is basically the set of techniques used to solve exactly that problem. If we do go ahead and solve this one, however, we get a trivial solution, right? So fortunately, my solution in the bottom left is actually correct, even though the matrix is all wrong. So if we say that S times V equals 0, and 0 here is vector like 0, 0, 0. Well, not just 0, 0. Okay. What we get is that -1V has to be 0, and -2V has to be 0, and V1 minus V2 is 0. Okay. And so basically V1 equals V2 equals 0. So we've obviously done something wrong. What have we done wrong? Well, we included the external environment outside the cell into our assumption of steady state. And I said earlier, like we can't do that because then the cell would be doing nothing, right? We're going to assume it doesn't change just because it's really big and we're going to ignore it. But we can't include the concentration of basically the external environment in the computation. Okay. All right. So we have to have even if we're not modeling the concentration of these things on the exterior, we do have to model the reactions that bring them in the cell and then bring the waste products out of the cell. So that's kind of those are the exchange reactions, and those define our boundary conditions. So in general, we'll have a nutrient uptake. So we'll have some molecule, let's say glucose, on the outside of the cell, and then we'll have a reaction that says, oh, we're going to convert that to glucose inside the cell. And then we have some waste product, let's say CO2, inside the cell. And then we'll have a reaction that specifically converts that to CO2 outside the cell. Okay, so as long as we include those fluxes in the model, then we can avoid these trivial solutions that sort of make sense. If nothing is changing, then the solution to the problem is that like every reaction is zero, right? Zero is certainly a valid solution to the problem, but it's not going to be optimal if your objective function is maximizing ATP production or maximizing biomass or growth, right? Okay, so ooh, yeah, I almost got this one right. My V vectors V1 through V4, it should be V0 through V3, but everything else on this slide looks correct. So here I've explicitly put in a V0 bringing A into the cell, and then a V3 bringing C outside the cell. And as long as I include those reactions, and I'm not going to include those A outside the cell or C outside the cell as metabolites, I'm just going to include their reactions. Now I can model the problem, and I won't get a trivial solution. In fact, the solution I'll get is that V1 has to be equal to V2, and that makes sense. Because if V1 was faster than V2, then the concentration of B would start to increase, right? And if it was slower, then the concentration of B would decrease over time. Okay, so if it's just one linear pathway, then if it's a linear pathway and there's no change in the concentration of intermediates over time, then the only solution is that the rate of each step of that linear pathway, they have to be equal, right? Same amount going in as coming out. Okay, so what does it look like if we make one of these models? Like, how good is the state of the art? This is a paper, I think it's from like 10 years ago, but I think it's still probably close to the state of the art. And this is a particularly well-studied strain of E. coli. And this is from the Paulson lab. They're one of the like leading labs that does this flux balance analysis. And they tried to make, at least at the time this was published, like the most complete genome scale model of a metabolic network. And what they did to test it is they took this E. coli and they grew it on a number of different conditions where they knew what was in the growth media. And then they also took different mutants from the KO collection. And the KO collection is a set of knockouts of every single E. coli gene that's not essential. Okay, so if it's essential and you knock it out, then you can't grow the cell. But for all the genes that you can knock out, they have them, and you can order this collection, and people do experiments on it and try to use that to deduce the function of different genes. So they went through the KO collection and identified the knockouts that were part of their metabolic model. And so they combined the knockouts with different growth conditions and came up with a set of, I think it was like 14,000 different experiments, that either had a knockout or a growth condition, or I guess both. And then they predicted not how fast it would grow, but like whether it would grow or not. And so this is that 2x2 matrix showing kind of how well the predictions worked. And you can see that it worked pretty well. It's certainly much better than random, but it's definitely not perfect, right? So it looks like there's a tendency that the computational model predicts that things will grow. And so of the things that the computational model said would not grow, most of them didn't grow, right? And of the of the things that the computational model said would grow, you know, most of them did grow, but you know, there's it's definitely not perfect, right? Because the baseline for this model is just predicting that everything grows, because most of them do. And this beats that performance, but it's far from perfect, right? So most of the things that did not grow were missed by the computational model. Like of the non-growers, it predicted like maybe a little less than half of them. So, but it's and then, you know, you can go deeper into, you know, trying to figure out, oh, which of those non-growth conditions and knockouts were sort of trivial, where a person who didn't know anything about modeling, but was just looking for like really important steps in like glycolysis or like enzymes that are present everywhere, how good would your performance be there? And, you know, it might be comparable. So that's why I say these models are very, they're very elegant and very powerful, but, you know, there's still a lot more complexity to the actual biological simulation than is captured by modeling these fluxes and assuming that things are optimal. Okay, we've only got a minute left, so what I might do is I don't want to go super quickly over the stochastic modeling, because I think it's really cool. And how many of you guys have seen the Gillespie algorithm? Okay, super important algorithm for all kinds of things. So I'm going to leave this, and we'll start with this next day. And then next day, we'll talk about some of the computational techniques and problems that we need to interpret metabolomic data. Okay, all right.